Hello, my name is Professor Hudson, and you're about to view narrated PowerPoint lecture on Chapter 13, Section 13.3. We'll continue with our discussion of how messenger RNA is translated into a protein. Let us begin. This slide is showing translation of a messenger RNA into protein. As mentioned previously, the first stage is initiation. This is where the ribosome assembles onto the messenger RNA and the first few tRNAs are brought in. The first tRNA is brought in by looking for the initiation codon or the start codon in the messenger RNA. And then elongation, the ribosome moves along the messenger RNA reading sets of 3 in order to bring in the tRNA with the correct amino acid. For this to happen, the anticodon on the tRNA must hydrogen bond complementarily to the codon in the messenger RNA. And then in termination, a stop codon is reached in the messenger RNA, which causes the ribosome to disassemble and release the protein. Let's look at each step in more detail. Step 1 for translation, initiation. Translation begins when a tRNA and messenger RNA bind to a ribosome. A pre-initiation complex forms consisting of the small ribosomal subunit and methionine tRNA and several other proteins. The UAC anticodon of the methionine tRNA in the pre-initiation complex binds the messenger RNA molecule by base pairing with the AUG start codon at the beginning of the messenger RNA. The large ribosomal subunit attaches to the small subunit, holding the messenger RNA between the two subunits and holding the methionine tRNA in its first tRNA binding site. The ribosome is fully assembled at this point and translation is ready to begin. Let's watch an animation of just initiation.
Translation initiation is a process in which mRNA, initiator tRNA, and small and large ribosomal subunits associate with each other to form a complex. Initiation of translation in bacteria begins when the 30S or small ribosomal subunit binds to the mRNA near its 5' end. This process is facilitated by a Shine-Dalgarno sequence in the mRNA, which is complementary to a component of the small ribosomal subunit called 16S ribosomal RNA. Though a shorter sequence is shown here, the Shine-Dalgarno sequence is actually 9 nucleotides long. Initiation factor 3 also facilitates the binding of the mRNA to the small ribosomal subunit. Once the 30S subunit IF3 mRNA complex has formed, initiation factor 2 binds to the complex and promotes the binding of the initiator transfer RNA for N-formal methionine to the complex. IF2 and IF3 hydrolyze GTP as an energy source to promote the association of mRNA, tRNA, and the ribosomal subunits. Translation initiation is completed when the large ribosomal subunit binds and IF2 and IF3 are released. Later, when translation is completed, IF1 is needed for the dissociation of the complex.
Now let's take a look at the second part of translation, which is elongation. During elongation, amino acids are added one at a time to the growing protein chain. A second tRNA anticodon base pairs with the second codon on the messenger RNA. As the tRNA is bound to the second tRNA binding site, the catalytic site of the large subunit breaks the bond, holding methionine to its tRNA, and forms a peptide bond between this amino acid and the amino acid attached to the second tRNA. The empty tRNA is released and the ribosome moves down the messenger RNA one codon. The tRNA with the growing amino acid chain is now in the ribosome's first binding site, and the second binding site is empty, awaiting another tRNA. A third tRNA with an anticodon complementary to the third codon of the messenger RNA enters the empty second binding site of the ribosome and base pairs with the messenger RNA. The catalytic site on the large subunit now passes the growing protein chain on the third amino acid. The empty tRNA leaves the ribosome and the ribosome shifts to the next codon on the messenger RNA. This process continues one at a time until a stop codon is reached. Now let's look at an animation of just elongation.
Chain elongation begins with the binding of a tRNA, which recognizes the next codon in the mRNA to the A site of the ribosome. This is catalyzed by the EFTU transcription factor and requires the hydrolysis of a GTP. Once the tRNA binds in the A site of the ribosome, the polypeptide chain is moved from the tRNA in the P site to the amino acid attached to the tRNA in the A site. Peptidyltransferase, a protein RNA complex present in the 50S ribosomal subunit, catalyzes the formation of this new peptide bond between the amino acids. The ribosome then translocates to the next codon. This process is promoted by elongation factor G and requires another GTP. This places the empty tRNA molecule in the E site of the ribosome and moves the tRNA containing the growing polypeptide chain in the P site. The next codon in the mRNA chain is positioned in the A site. The uncharged or empty tRNA in the E site then leaves the ribosome and a cycle of chain elongation is completed. Through subsequent cycles of chain elongation, the polypeptide chain continues to elongate one amino acid at a time.
The last stage in translation is termination. Termination occurs when a stop codon signals the end of translation. When the ribosome reaches a stop codon in the messenger RNA molecule, releasing factors cause it to release the completed polypeptide chain and the messenger RNA to disassemble into its large and small subunits. A tRNA is not brought in when the stop codon is reached. Let's watch an animation of this.
Termination begins when a stop codon appears in the A site. Since there is no tRNA corresponding to the stop codon, a release factor binds in the A site. The binding of the release factor causes the polypeptide chain to be cleaved from the tRNA. The polypeptide is released, and then the tRNA is released. In the last step, the two ribosomal subunits and the mRNA dissociate from each other. This completes the termination process.
This slide is showing the elongation of a polypeptide. Remember, the elongation is the second step in protein translation. Basically, the anticodon in the tRNA must complementary bond to the codon in the messenger RNA. This facilitates bringing in the correct tRNA with the correct amino acid. Now let's look at translation, the complete process, one more time.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA, paired with the previous codon, is passed to the P site and then to the E site, as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
Summing up, decoding the sequence of bases in DNA into the sequence of amino acids in proteins. Each DNA gene codes for a single protein. Transcription produces a messenger RNA strand complementary to the DNA gene template strand. The messenger RNA strand leaves the nucleus and associates with a ribosome in the cytoplasm. Transfer RNAs in the cytoplasm are loaded with their appropriate amino acids by cytoplasmic enzymes. The ribosome selects the tRNAs on the basis of the base pairing of the anticodon with the exposed messenger RNA codon. The messenger RNA contains a stop codon to define where protein synthesis ends. So the central concept of genetics, or the central dogma, begins with our DNA, and this DNA contains the information necessary for making a protein. An enzyme called RNA polymerase is going to read the information in DNA, and it can read on either strand of the DNA in order to produce a messenger RNA molecule, which is a snapshot of what's in the DNA. After processing, this messenger RNA will leave the nucleus and go to the cytoplasm. The messenger RNA, in conjunction with ribosomes and tRNA, will form the protein. The process of making messenger RNA is called transcription, and the process of making the protein from the messenger RNA is called translation. Question 12. If the messenger RNA transcript A, U, G, C, G, C, U, G, C, A, U were to leave the nucleus and undergo translation at a ribosome, what would be the sequence of the tRNA anticodons translating this nucleotide into protein? Choose the correct answer. This slide is a summary of gene expression in eukaryotic cells. This slide shows from start to finish the process of transcription of DNA into messenger RNA and the translation of messenger RNA into protein. Let's watch one final animation summarizing the entire process.
Sequence of nucleotide bases in DNA carries genetic information in units that are called genes. Structural genes encode the information for specific proteins. To create a protein, a gene must first be transcribed into a sequence of nucleotide bases in a messenger RNA molecule. The messenger RNA sequence is then translated into an amino acid sequence of a protein. This sequence of amino acids in a protein molecule determines the shape and chemical characteristics of the protein. Thus, each structural gene specifies a specific protein in the cell that carries out a specific function based on its chemical characteristics and molecular shape. This function of the specific protein gives the cell and the organism the specific trait coded by the gene.
And this concludes our narrated PowerPoint lecture for chapters 12 and 13.